Hello everyone. In today's lecture, we'll study about different spectroscopic techniques and um, different techniques used to study the interaction between enzyme and substrate. In the previous classes, we have been studying about enzymology, the history of enzymology, and how to identify genes related to enzymes, and then uh, find out those genes either from known databases or make a cDNA library or a gene library in order to identify the gene encoding those enzymes, and then fortunately cloning them into uh, different vectors like bacteria and animal vectors, followed by the expression of those enzymes in different hosts. And then we also studied how to like express those different enzymes in host, and how the enzymes catalyze different type of metabolic reactions also in the previous class. In this class, the major focus is on enzyme substrate interactions and how these interactions can be detected by various spectroscopic and other techniques which we'll be studying today, which includes electrophoresis techniques and other chromatographic techniques, which we have already few chromatographic techniques which we have already studied in the previous classes as well. To begin the enzyme substrate reaction, so we have enzyme plus substrate forming a enzyme substrate complex, and this enzyme substrate complex can be further converted to enzyme product complex. There could be some changes in the substrate which will then there will be some changes in the substrate that will convert the substrate into product, and finally this enzyme product complex um, will give us enzyme and product. Okay, and this enzyme can further convert other substrate molecules um, into the enzyme substrate complex, and this reaction continues where we have maximum product formation. So this is the enzyme substrate reaction, and there could be many interactions which occur in enzyme and substrate, and this interaction could be um, it could be hydrophobic interactions which are very important. It could be hydrophobic interactions, it could be ionic interactions, it could be other non-covalent interactions like salt bridges and dipole interaction. So these are the different interactions which occur in between enzyme and substrate to get enzyme substrate complex. And because of this reaction, we have specificity. We have specificity of enzyme towards substrate, and this specificity can be divided again into three components, which is because the specificity could be because of geometric complementarity, it could be also because of electronic complementarity, and it could be also because of stereo specificity. So these interactions are majorly categorized into um, uh, these three categories, where geometry complementarity means the overall shape of enzyme and substrate and how they complement each other. Electronic complementarity refers to the ionic or different positive and negative interactions which could complement the enzyme substrate complex formation. And stereospecificity is that the L type of amino acids in enzymes react specifically to there is a stereospecificity towards the substrate. So these are the different types of complementarity which occur in during enzyme substrate formation. And this enzyme substrate complex, these interactions could be we could study them because these interactions, the formation of enzyme plus substrate, the formation of this enzyme substrate complex, or any interaction, it could lead to changes in size, which could be studied by electrophoresis. Changes in size can also be studied by gel filtration chromatography, which we already studied in the previous classes. And there could be other hydrodynamic changes which occur. So these hydrodynamic changes again could be also studied by gel filtration. Also there could be change in the surface chemistry, that is the surface chemistry from hydrophilicity could change to hydrophobicity. There could be changes in the surface chemistry, that is there could be loss of charge, it could be less negatively charged or less positively charged, and these can again be studied by ion exchange chromatography, which we already studied in the last chromatographic class. And finally, there could also be free energy, change in free energy, or there could be change in the overall enthalpy of the so whenever there is formation of this uh, enzyme substrate complex, it could release enthalpy, or it could be either exergonic process or endergonic process, that is it could either release heat or take up the heat. So all these heat related changes, there could be a change in free energy or enthalpy, or there could be change in the heat, and this could be measured by ITC, that we will study in today's class. What is this ITC? We'll study this. So so in the electrophoresis, again electrophoresis could be so we have already studied this gel filtration ion exchange chromatography. So electrophoresis, again, to study the native protein, we have native page. Okay, we have native polyacrylamide page, and to study denatured, or in order to study denatured proteins, or so in order to study denatured protein, we could have urea page or SDS page. Okay, so there are two types of electrophoresis. There is a denaturing electrophoresis and there is non-denaturing electrophoresis. So depending upon the choice, we can either do a native page or urea page. So this is the example of one of the interaction in which this is a modified version of ion exchange chromatography. So here we are checking for an enzyme which has a affinity towards DNA. So we have an enzyme we are checking for an enzyme that has affinity towards DNA. And so we have this column packed, column which is packed, to which we'll add DNA, and this DNA will non-covalently go and bind to the beads. Okay, so there will be binding of DNA first. The first step is binding of DNA. It will go and bind to the beads, and to this packed column, we'll add a protein X or enzyme of our interest, which has, in order to check the affinity. So whichever protein or enzyme does not have affinity for that DNA, it will get eluted or washed out. Whereas this DNA protein or DNA enzyme mixture could be again run on SDS or agarose gel, and we could see that it could tell us that how much of this protein is bound to this DNA. We could get different elution fractions. So this is our first fraction. This is our first fraction. This could be second, third, fourth. So we can have different fractions, and that we can run on the gel. So we got a maximum intensity band in the third fraction. So we can say that the protein is getting the maximum protein or enzyme is getting bound in this third fraction. So similarly, this is how we can separate. So this protein will have some interaction. It will have some ionic interaction with the DNA, and this is how we are exploiting ion exchange chromatography in order to study this. Similarly, we can use gel filtration chromatography also to check the hydrodynamic changes, and it follows the same principle. The gel filtration chromatography also follows the same principle. Then we have electrophoresis technique. So in the electrophoresis also, as I mentioned before, that so electrophoresis, so we have a gel. It could be either denaturing gel or non-denaturing gel. So in non-denaturing gel, we do not add denaturing agents like urea or SDS. So SDS, urea and mercaptoethanol are all denaturating agents. So in a native page is used basically to study the suppose our protein, to study the native conformation. So what is this native conformation? Native conformation refers to the conformation of protein which exists in the solution. Okay, so if a protein is folded in such a way in the solution, okay, so the native page, the whole protein will run in this conformation only. There won't be any change in the conformation of the protein. So because there are no denaturing agents like urea, SDS or beta mercaptoethanol. So native pages are used to run when we want to maintain the overall native conformation of the protein. So from here so here on the left hand side we run the ladder. So we have ladder run, and then we can load the samples here native in order to do a native page. Whereas in the urea page, urea is a denaturing agent. So it will so if this protein was a dimer, okay, if this protein was a dimer and we got a band here, okay, we got a nice band at 200 kilodalton band, okay, in the native page. The same protein, if we run in the urea page, will get a band at it will run lower. It will get a band at 100 kilodalton. Okay, so it's a dimer of 200 kD, 200 kDa, 200 kilodalton. So in the native page, so conformation will be retained, whereas in the urea page, it will get denatured, and then hence we'll have two monomers getting forming a band here at 100 kilodalton. So if this is our ladder, here is 200, we'll have a 100 kilodalton band here. Okay. So next we can measure enzyme substrate interactions in UV visible spectroscopy. So what is this UV visible spectroscopy? So we have wavelength. So UV region falls from, as you can see from this picture, UV region, this ultraviolet region, okay, it falls from, UV region falls from 190 to 400 nanometer. Okay, UV region falls, and then we have a visible region which begins from 400 to 780. Okay, so we have a ultraviolet region here, which falls between 190 and 400, and then we also have far UV and near UV. So far UV falls from 190 to 250. Okay, it is UV again is divided into far and near UV falls from 250 to 400. Okay, so and then we have a visible region. Which begins from 400 to 780 nanometer. So so what happens when we have some molecule in the ground state, whenever a light or energy falls on them, whenever we have light falling on the molecules which are in ground state, they get excited from the ground state. So after getting excited, they could have a sigma to sigma star transition, and this sigma to sigma star transition, okay, this transition mainly occurs at single bond, that is alkanes. All the alkanes have this transition, whereas alkenes, whenever there is double bond, that is we have pi to pi star transition. So this transition occurs in the double bonds. Okay, and so we can measure this transition, because how we can measure this transition, as whenever there is transition from sigma to sigma star bond, we have a lambda max. We have a lambda max at 150 nanometer. Okay, we have lambda max at 150 nanometer. That is, we can measure the absorbance. At 150 nanometer. Similarly, for alkenes, whenever there is pi to pi transition, this transition, pi to pi transition, and we have a lambda max at 170 to 190 nanometer. That is, we'll get some absorbance peaks. Like this, okay, for different transitions. So whenever if we have double bond characteristic in our enzyme, or if there is a double bond characteristic in enzyme substrate interaction, if there is a formation of a double bond, we'll have a lambda max at 170 to 190 nanometer, or if there are all the aliphatic carbon present, we'll have an absorption at 150 nanometer. So this absorption spectroscopy or UV visible spectroscopy, we use this to measure the absorbance. We use the Beer-Lambert's law. Okay, and this Beer-Lambert's law refers to absorbance is at epsilon Cl, where epsilon is molar extinction coefficient, C is the concentration, so C is concentration, L is the wavelength, and this is the molar extinction coefficient, and this is the absorbance. So UV visible spectroscopy is of two types. So this is a single beam. This is a single beam UV visible spectrophotometer. This is a single beam UV visible spec. So what do you mean by single beam? So we have a source of light. It could be either visible or UV. So this is a UV visible. It will have both sources, visible light source and UV light source. Okay, we'll have a mirror, and we'll have a monochromator. So this monochromator will filter out all the wavelength, and it will only pass the wavelength of our interest. Okay, so we can set the monochromator to either of specific wavelength. Let's say we could set it as 450, or we could set it at 600. So it will allow only that particular, like it will allow only 500 or 600 wavelength, or we could have a range that is from 400 to 500. It will allow. So we have a reference cell and we have a sample cell, and then we have a detector. So we have a single beam going into reference cell, and the and different beam will go into the sample cell. So we have a single beam here which goes into the detector, and it will show us some absorbance which is directly proportional to incident light, and this is for the reference. And similarly for the sample, we'll also get some absorbance, and then we have to compare these two absorbance, like how much the absorbance has changed for the sample, or how much the absorbance has changed for the enzyme substrate reaction. And the reference cell, we could add only the substrate without any enzyme. So we can check what are the changes in the absorption. Whereas in the double beam UV visible spectrophotometer, what happens? So this is a double beam. Why is it a double beam? Because we have a monochromator. So till here it is same, but when here the light is getting divided into two beams. So we have this one beam going into the detector, and then so we have a reference cells. Okay, so one beam going into reference cell, and the second beam which is divided by this mirror. Okay, the second beam, so basically this mirror is splitting the one beam into two, and the other beam is going into sample. So here one beam is splitted into two, which reduces the error, whereas here we have a reference cell, then we again have to remove the reference cell, and have to again check the absorbance for the sample. So we'll have a margin of error occurring here as compared to this. So this is the advantage of having a double beam spec. And overall principle remains same. Here also we'll apply Beer-Lambert's law. So this is the Beer-Lambert's law here. As you can see that this is the total area, and it is covering the length L. Okay, the distance, this distance is a path length, that is the L here in the Beer-Lambert's law, in this Beer-Lambert's equation. And then we have absorbance. So we'll have a absorbance of the species, which we'll put here, and epsilon is the constant which will depend on the solution, and concentration again is the factor that will depend upon the solution. So this is the concentration here that will depend upon the solution. So next we can also measure isothermal, we can measure the enzyme substrate interaction by ITC. So what is ITC? ITC is isothermal titration calorimetry. So the basic principle behind this is that we have the basic principle behind this is whenever there is interaction between enzyme and substrate, Okay, whenever there is a complex formation between enzyme and substrate, there could be either release of heat. Okay, there could be a release of heat, or it could be either absorption of heat. Okay, so the process could be either exergonic or it could be endergonic process. Okay, so and this heat which is released can be collected, this heat which is released can be measured by this ICT instrument. So this ICT instrument has a reference cell and it has a sample cell. So in the sample cell we'll feed our enzyme and substrate, whereas in the reference cell we can add only our substrate, and here we can add enzyme plus substrate. So so then we titrate it. So by titration I mean we increase the enzyme concentration. Okay, we gradually increase the enzyme concentration is gradually increase. Okay, so there is an increase in the enzyme concentration, and whatever interaction, so as we increase the enzyme concentration there will be more interaction with the substrate, and as there is more interaction with the substrate there will be more heat which is released because of these interactions. Because of the small interaction there could be release of heat. So this heat is measured here on the y-axis using the so this isothermal titration calorimetry has a probe to measure the heat, and this measured heat is plotted against the, it can be either plotted against time, like how the stoichiometry is changing along with the time, or the molar ratio. So with this we can also calculate Kd, that is the dissociation constant. Kd is dissociation constant. Okay, so Kd is the dissociation constant here. It will tell us, so basically this is how, so as we increase the enzyme concentration, this is how our dissociation curve will look like. Okay, so what are advantages of this ITC method? So advantages, so it's a very sensitive method. So it helps us to calculate the stoichiometry, and also it will help us to calculate Kd dissociation constant. Okay, and then we there is no need to have any no need of any chromophores or fluorophores. Okay, it's just measuring the interaction between enzyme and substrate. The another advantage is that it interacts in the solution. So whatever the enzyme substrate activity interaction buffer solution which we have, it will interact in that solution only, and it is very quick, it is very fast and efficient method to study the enzyme substrate reaction. The disadvantages are that the disadvantages here are that it requires very large amount of sample, and it is non-specific. So if our solution is contaminated with other substrate, or if we are using some wholesale lysate, or there could be other cellular fractions which are contaminating, or which are interacting with our enzyme, it will give us the heat the different different readings for those, and it will induce more error. So and it is also low throughput, that is we cannot run multiple samples at same time. Okay, we can do this only one sample at a time, and it is kinetically it is very slow. So those reactions which are kinetically slow, only those can be uh, studied with the ITC method. Okay, so these are the advantages and disadvantages of this method. The next method is SPR, also known as surface plasmon resonance. So as the name suggests, so we have two things happening. So we have a surface plasmon, and then we have a resonance phenomena which is observed. So this method is also surface sensitive. It is surface sensitive, and it could be high throughput. Okay, so ITC was not as high throughput, but this method could be high throughput if we want. So it basically measures the surface plasmon. So we have a metal surface. Let's suppose we have a metal surface, any gold. It should be a gold or heavy metal, not heavy metal. It should be a metallic surface. So all this metallic surface will be covered in the electrons. Okay, this surface will be covered in electrons. Okay, and this electrons will have a charge on them. They will have a charge on them. Okay, this surface will have a charge on them under a magnetic field. Under some influence, it will have a charge on them. So this charge will resonate like this in form of waves. Okay, so there will be a resonance of charge in form of waves. Okay, in presence of some analyte, in presence of a analyte. So if we have a analyte solution, so if we have a analyte solution surrounding this this metal surface, okay, there will be a change in the resonance. Okay, there will be a change in the resonance, and this can be captured by, so if we have a light source, if we have a light source and we have a light reflecting here, okay, so there will be a reflecting light at so here this is the incident light at theta, angle theta, and there will be a reflecting light. Okay, so if we add some analyte, or if the refractive index of this solution changes, okay, this theta will change. So this theta will also change because of the refractive index of this solution. Okay, and why will the refractive index change? Suppose we have a solution of substrate, okay, of substrate only, and it has some refractive index 5. Let's say it has some refractive index 5. When we mix enzyme and substrate together, there will be change in the refractive index. Okay, this refractive index will increase or decrease. It could either become 1 or it could either become 10. So there will be a change in the refractive index, and this change in the refractive index will lead to the difference in this theta angle, and this is what we are measuring in the surface plasma resonance. Okay, so we have intensity graph here, and we have an angle theta which we are measuring here. So this is the light without any with only substrate. Okay, and when we add enzyme, there could be decrease in the angle like this. There could be decrease in the angle, and this is captured by this instrument. Okay, and this angle could either be a associative angle. It could be either because of association between enzyme substrate, or it could also capture Kd also because of dissociation between enzyme and substrate. So this is how the overall setup of the SPR looks like. So we have our analyte solution. Okay, analyte solution. So this is the solution where we add substrate or our biomolecules. Okay, and suppose this analyte is binding to the biomolecule, the protein is binding to the receptor molecules here, and then we'll have a plasmon wave which is generated by this metal surface. Okay, and below this we have a solution. So the light source, okay, we'll have a light source, and then we have a detector. So whenever there is a binding of analyte to this receptor, there will be a change in angle, there will be a shift in intensity. So this is a black line without the binding of the analyte. Whenever the analyte is binding, there is a shift. Okay, there is this shift, this theta, there is a change in theta. Okay, this theta is changed, this theta is now decreased, and this shift in intensity is measured using the this is the graph what it looks like. So this is a good graph where you are getting a good saturation. Okay, and then you have a decay, or there is a dissociation of the both analyte and the biomolecule, whereas here it's a bad because there is no saturation which is observed. So then we can also study different association and dissociation kinetics of the surface plasmon resonance. So let's study the association kinetics of SPR. So we have a surface. So we have A molecule and we have B molecule, and then we have Ka will be A bound to B, whereas Kd will be dissociation of this bound AB to A plus B. So so we can calculate this dr by dt, where R is analyte, rate of binding of analyte with respect to time, analyte bound. Okay, so this rate of bound analyte with respect to time is given by Ka, that is association constant, R max, that is maximum analyte bound to the biomolecule, upon minus R upon Kdr. And we can also write this in form of concentration in terms of concentration also. Ka, where C is the concentration of the protein which is our analyte which is bound. R max is the maximum analyte bound, minus Kc plus Kd. So this is the association kinetics equation for SPR. Similarly, we have dissociation kinetics equation as well for SPR. So for dissociation kinetics, we have the rate of change of bound analyte, okay, is directly proportional to Kd R, okay, minus because it is decreasing. So if we calculate this, if we convert this, so if we take a log, so here R0 is the initial response time where the analyte is bound. Analyte is not bound, okay, it's a initial. So this is how the graph will look like. So we have taken log, that's why we have a straight line. Okay, and then here we have Rn R0 upon RT, where RT is response or analyte bound at time t. Okay, and the slope here will be equal to Kd. Okay, and then we have time in seconds on x-axis. So from here we can calculate Kd, that is dissociation constant. Okay, so what are the different applications of SPR? SPR will help whenever we want to calculate different dielectric properties of enzyme substrate reaction. Um, so this could be high throughput, as in biologically it could be used to as a biosensor, or we could study antigen antibody reactions or protein ligand interactions, and also used in tissue engineering. Okay, so it has various applications. So with this we move to the assignment questions. The DNA binding proteins or enzymes eluted from chromatography column by. So we can elute DNA binding proteins or enzymes from chromatographic column by applying a gradient of increasing. So we have some interaction. So we have a DNA and we have a protein bound to it. Okay, so this DNA and protein are bound by ionic interactions, and these interactions can be easily eluted by if we increase the ionic strength. Okay, if we increase the ionic strength. Okay, so if the binding occurs at 1 molar NaOH, we can just increase the ionic strength, or we can keep on increasing from 1 to 2 molar or 5 molar, and it will eventually elute out. Okay, it will eventually elute. So by applying a gradient of increasing ionic strength, or ionic strength of the solution can elute the enzymes from the DNA binding proteins. The eluted fractions in DNA binding proteins or enzymes from chromatographic technique are analyzed for the purpose of studying the binding properties of the separated enzymes. So in order to study the different binding properties, okay, size, it won't be help to tell the size of the protein which are bound to the DNA. That will be checked after doing electrophoresis. pH and concentration are also not possible to check, so we can only check the binding properties of the enzymes and enzyme proteins to bound to DNA. So the correct answer is D. The correct answer is D. Migration of proteins in SDS page primarily depend on their net charge, solubility in gel matrix, pH at gel interface, enzymatic activity. So this depends on the net charge. Okay, so what is the net charge on the SDS page? So it will it depends on net charge, and it also depends on the size and overall friction or tension between the molecule and the SDS. Okay, so it will depend on net charge, size, and the overall friction. It's independent on pH, enzymatic activity, or the solubility of the gel in the matrix. The amino acids that dominate the absorption spectra near UV region of proteins are, so near UV region we have maximum absorption of aromatic amino acids, and the aromatic amino acids are sorry, not aromatic amino acids. We have at near UV region we have tyrosine and tryptophan. Yeah, so tyrosine and tryptophan both are aromatic amino acids. So we have aromatic amino acid here. So this is tyrosine, and tryptophan also has similar structure. Okay, tryptophan also has a aromatic structure. So the aromatic rings of tryptophan and tyrosine, they will absorb at near UV region. The double beam spectrophotometer improves the measurement reproducibility by, so there it basically it will reduce the errors. Okay, so by compensating the fluctuation using the reference cell. So it will compensate the whatever fluctuations or errors we are getting by the reference cell. So it will take care of that. Reducing the intensity of the incident, no. The intensity is reduced by a ND filter, or ND filter, or we have a neutral density filter that will reduce the intensity. Using multiple detectors for increased sensitivity, no. Exposing the sample to multiple wavelengths, no. This, so the correct answer is D. Additional insights that can be provided by ITC beyond binding affinities is reaction kinetics, molecular weight, structural details of biomolecules, mechanism underlying molecular interactions. So yeah, the answer is C. So from ITC we can identify the basis of molecular interactions also. Okay, so whether they are yeah, so whether different mechanism of interactions like association and dissociation, or how they are interacting, okay, at molecular levels. So as the titration is gradually performed, the ITC measures the heat changes. Okay, so it essentially measures the heat changes. This pH, optical density, concentration, it does not measure. ITC quantify the binding strength between the molecules by, it measures the equilibrium constant, that is Kd. So the answer is D. Measuring the concentration of ligand, calculation temperature changes during the binding, analyzing the change in entropy. So so it will it basically measures the it measures the binding constants, that is Kd. SPR is utilized in determining the binding kinetics between an antigen and antibody by quantifying the changes in the refractive index. So there will be a change in the refractive index, and thus it will change the angle, it will change the theta, which is of the light which is refracted, and the angle of theta will change, which will tell us about the different kinetic interaction, binding interactions. The kind of information that can be obtained through SPR in structural and interaction studies is affinity and dissociation constant, temperature variations. So basically we can check the affinity and dissociation constant from SPR. Okay, there we can't measure change in pressure, temperature and molecular weight variations. Dash lamp is used as a source of visible light in UV visible. So for visible light we generally use tungsten lamp. So answer is B, tungsten. Which of the following is also called electronic spectroscopy? So, electronic spectroscopy is nothing but UV visible spectroscopy. The substrate specificity in enzyme that determines whether to bind with an L type or D type of substrate in terms of sugar. So, this L type and D type is because of stereospecificity. The correct answer is stereospecificity. Which of the following have the lowest wavelength on electromagnetic spectrum? So, lowest wavelength is for gamma rays. Okay, they have lowest wavelength on the electromagnetic spectrum. Yes, so